synaptic vesicles. First one on this is physiology and histology. So, synaptic vesicles are preformed storage vesicles for neurotransmitters, like a storage pool, and calcium ions. Hey, wizard. Activates the snare complex to form between vesicles and the cell membrane. And then the vesicle docks with the nerve terminal cell membrane to send those neurotransmitters in order to cause a synapse. And those neurotransmitters are released by exocytosis. And then the membranes recycled after coating with clathrin. And then the vesicles are reloaded with neurotransmitters. Yeah, I'm live at school. I'm just in a study room. So the neurotransmitter is released from receptors, and at the end of the signal, there's three things that happen: diffusion. No, it's just me in the room. Yeah. But at the end of the signal, diffusion happens, reuptake through presynaptic reuptake pumps, and then degradation of specific enzymes. Something like that. I don't know, this is all pretty weird, complex stuff. Okay. There's many things that can hamper synaptic transmission, like acidosis or alkalosis, stuff like that. Botulism, that can also hamper it, because it's affecting the neuromuscular system. It can cause respiratory failure, and that's what causes death when people when any animal gets botulism. Some animals aren't as badly affected. But then there's there's many kinds of different receptors and terminologies that are used in Mario for these receptors, like a ligand. A ligand is something that binds to something else. An agonist is something that stimulates a receptor, and an antagonist is something that blocks anything attaching to receptors. Yeah, there's all sorts of live streams out there. There's also postsynaptic receptors. There's ionotropic ones, which are very fast signaling, and they can be excitatory or inhibitory. We've kind of already talked about those a little bit. And then there's metabotropic, which are G protein coupled. And these can also be excitatory or inhibitory. And a little bit more complex than that, but I'm just going over them briefly. Then there's many different kinds of neurotransmitters. monoamines, acetylcholine-like things like small molecules, amino acids, peptides. Here are some examples. Acetylcholine, that's just a small molecule. Dopamine, norepinephrine, epinephrine, serotonin. I'll take away the acetylcholine. These are monoamines. Glutamate, aspartate, GABA, glycine, those are amino acids. Substance P and opioids, those are peptides. And then there's endocannabinoids. That's another kind of neurotransmitter. But yeah, everyone's probably heard of dopamine or yeah. Everyone's probably heard of dopamine or epinephrine. And those used to be called adrenaline or noradrenaline. Still are. Just those were the older names. It's okay. Slow morning. As long as I get to talk to some people, I'm alright. Yeah, this ain't for me. Right now it says 7. That's what I'm seeing. But I don't know, sometimes. Hey Thomas. Sometimes it says something different. I'm doing okay, Thomas. How are you? Anyway, acetylcholine. It signals at the neuromuscular junction, at the enteric nervous system, at the parasympathetic nervous system, at the central nervous system. And neurons that release ACH are known as cholinergic neurons. I'm not really working. I'm just studying. I've actually got an exam at 8 a.m. It's 6:38 right now where I am. But yeah, I'm just trying to teach people and teach myself, I guess. You know. Acetylcholine has to terminate its transmission as well. These are all parts of the synapse process. The most common thing that breaks down acetylcholine is organophosphates, which are a type of chemical insecticide. Not good for us. Okay, and then there's also drugs or toxins that bind and antagonize acetylcholine, like atropine, antihistamines, like diphenhydramine, or diphenylhydramine, I guess is the full name. Antiemetics, like scopolamine. I've never heard of that one before. But you guys may have heard of Benadryl. It's another word for, that's the brand name for diphenhydramine or diphenylhydramine. Okay, and then tricyclic antidepressants. And then some plants like belladonna or ginseng weed. These could cause blockage of acetylcholine receptors, which is not a good thing. Well, it just depends, I guess. It's not good if it's unintended. And then there's those monoamines again. adrenaline, dopamine, serotonin. Thanks, William. Catecholamines as well. Dopamine, norepinephrine, epinephrine. These are also called catecholamines because they have a catechol group. Yeah. And about 90% of the monoamines are taken back, excuse me, into the nerve terminal by a neuronal reuptake transport system. And only a small amount is taken up by post-junctional tissue and metabolized. And a lot of those metabolites are excreted in the urine. Yeah, I'm sorry. My laptop stream has a fan. Sorry about that, Joshua. Yeah. It's plugged in. I can unplug it, I guess, and then it won't have that loud noise. But then the battery drains pretty quick. But it's due to the fan in my laptop. That's why it's so loud. I'm sorry about that. Hopefully it's better now. But anyway. Alright. Some functions of dopamine: reward, motivation, mood, movement coordination, actually, hormone release, blood pressure, gastrointestinal motility, and renal blood flow, prolactin. Lots of functions of dopamine. And Parkinson's disease is actually a deficiency in dopamine transmission, and so therefore a lack of movement coordination. Serotonin, 80 to 90% of serotonin's in the gut. And the rest of it's in the CNS, central nervous system. It controls appetite, mood, anger. Yeah. Zoloft, Prozac, those are selective serotonin reuptake inhibitors. Medications you may have heard of. And then there's something called serotonin syndrome. If you have too much serotonin or you medically take it too much, like, with medicine, you can get jittery, anxious, confused. You might sweat a lot. And you might develop movement disorders, possibly. Okay, then there's glutamate and GABA. Those are very common neurotransmitters. Glutamate is excitatory, GABA is inhibitory. And they have a whole cycle that's very detailed, and I'd rather not get into. But thanks for watching, Wizard. Appreciate you. Thanks for tuning in. It's good to have you, man. But yeah, the neurotransmitters are very flexible. There's many receptors for them. Locations of those receptors are pretty much all over the body. Plenty of diseases associated with neurotransmitter dysfunction. Anyway, then the rest of this, we're kind of just getting into anatomy again, somewhat. Like, the central nervous system is comprised of the brain, which is the cerebrum, brainstem, and cerebellum. And then the spinal cord. Hey, Trinity. And then the brain and spinal cord are encased in dura mater, and they are protected by the blood-brain barrier. They're also encased in bone, like your brain is encased in your skull. Your spinal cord is encased in your vertebrae. Now, your vertebrae are not completely protected. There's plenty of foramina, which means holes where nerves escape, but it's very well regulated by the body. Then there's neurons, there's support cells, there's meninges, cerebrospinal fluid, the blood-brain barrier. Kind of already talked about a little bit of this. But the meninges is made up of 3 membranes that wrap around the CNS, which are the dura mater, which is the tough, inelastic cover, the arachnoid mater, which is the space between this layer and the pia mater, and it's filled with cerebrospinal fluid. And then there's the pia mater, which closely adheres to surfaces. Then there's glial cells. These are support cells, astrocytes, oligodendrocytes, microglia, ependymal cells, oligodendrocytes make myelin in the central nervous system, and Schwann cells make myelin in the peripheral nervous system. The microglia are basically macrophages in the brain and in the CNS. And then the ependymal cells are epithelial cells lining the cavities of the brain and spinal cord. Astrocytes are support neurons, basically. They're involved in the physical structuring of the brain, and they get their name because they're star-shaped. And, okay, that's all we got for physiology and histology. Moving on to immunology. We have let's see here. Okay. Let's see, 10, 27. There we go. Okay, so bacteria. Talking about bacteria. Plenty of different kinds of bacteria. They don't have a nucleus. And they don't really have other membrane-bound organelles. They've only got a single circular chromosome. And some bacteria have chromosomal genetic material outside. They have genetic material outside of the chromosome, like plasmids and stuff like that. Now, there's gram-positive and gram-negative bacteria, which just means it's either got a thick peptidoglycan layer or a thin one. There's plenty of ways to stain these bacteria in order to find out which one they are, like acid-fast staining. Yeah. Anyway. There's extracellular and intracellular bacteria, which means they grow inside or outside of host cells. There's obligate intracellular bacteria, which they have to reproduce inside the host cell. And then there's facultative, which means they can do both, but they invade host cells when they gain a selective advantage in the host. And not all bacteria and not all microbes are bad. We've got plenty of microbes in our gut and in our body, on our skin, all over. And exposure to those good bacteria, those good microbes, keeps us healthy. There's exotoxins and endotoxins. Different in a couple ways, and they are like one is highly antigenic, exotoxins, and then endotoxins are poorly antigenic. But the ones that's highly antigenic and highly toxic, exotoxins, there's vaccines for them. But endotoxins, which are not that toxic and poorly antigenic, there's no vaccines available. So it's kind of a good thing. Okay. MHC1 presents intracellular antigen fragments, and MHC2 presents extracellular fragments, so fragments from extracellular microbes. And it's happening all over the body, in the lymph nodes and in the mucosal associated lymphoid tissue, MALT. There's plenty of bacteria that fit these descriptors. Okay. I would say that's pretty much it for today. Sorry about the environment. I'm trying to be quiet because I don't want to disrupt my peers. But when I'm at my apartment, it'll probably be better. So thanks for watching, everybody, and I'll see y'all in the next one.